Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1B for June 2022. This is the part 2 video. I'll put the link to the first part and the third part below the description box. So here let's begin with question 4. It says, scientists collect data from a grassland ecosystem. For each trophic level, they determine the mean number of organisms in a square meter and the mean dry mass of these organisms in a square meter. They say the table shows the scientists' data. So this is where the information is. We can see numbers as well as their mean dry masses for each of the trophic levels. So the first question says, draw a labeled pyramid of numbers for this data. So we can see the producers are the most, so I put them at the bottom, and then the primary consumers, the secondary consumers, and finally the tertiary consumers. The next part says, describe how you could collect data to find the mean number of producers per square meter in the ecosystem. So we need to use a quadrat. And when you use a quadrat, you have to place it randomly throughout the whole area. And after, you have to count the number of plants per throw. So every time you throw, you count the number of plants in that quadrat. Then we have to repeat the calculation and find the mean number of producers per throw. Finally, we need to calculate the total sum of plants and divide that by the area in order to produce the mean number of producers per square meter as required. So here you would get 3 marks. Moving on, they say the mass of organisms at each trophic level is called the biomass. They say the percentage of biomass in the producers that is transferred to the primary consumers is 4.5%. They want us to calculate the percentage of biomass in the secondary consumers that is transferred to the tertiary consumers. I want us to go back here. We can see this is what is found in the tertiary consumers, and this is what we have in the secondary consumers. So it means this value here times as a specific percentage will equal to that. So let's go to the next page. That's why here I said X% of that is what gives us that. So we will be looking for the value of X, which is the percentage. So making X the subject, you get 2.4 times 100 divided by 10.6, which gives us 22.6%. Therefore, the percentage is 22.6. Next, they say comment on the energy transfer in this ecosystem. When they say comment, you have to write based on the information you see in the table. And they said in your answer, refer to the data from the table and the percentages of biomass transferred. So before I answer here, I want us to go back here and look. You can see the percentages transferred. We see a lot of mass was found in the producers, but when you go to the primary consumers, it's slowly decreasing, meaning some of this was not transferred to this trophic level and some of this was not transferred there, as well as some of this not transferred here. So I'm going to be giving reasons as well as talking about that part. So here I say, the top of the pyramid contains fewer organisms, so they contain less biomass. Energy is lost at each trophic level, so less energy is transferred to the next. Then I say the most energy is lost between producers and primary consumers. To see this, you can go back here and observe that there is a great percentage loss in energy, or energy transferred is really low. So we can see this is where we have the greatest loss from producers to primary consumers. And also I said energy loss occurs due to respiration. So in some organisms, as to why it's not taken to the next trophic level is because some of the energy is lost in respiration and not everything is eaten. Like for producers, sometimes the roots are not eaten, the branches are not eaten, and not all eaten parts will be digested. Most plant material contains cellulose, and if cellulose cannot be digested, the energy contained in that cellulose will not go to the next trophic level. The next part is excretion. So excretion leads to energy loss because some are lost as urine, some in form of sweat, so all that can lead to energy losses. So this brings us to the end of question 4. Let's move on to question 5. Question 5, the diagram shows a wind-pollinated flower with some structures labeled W, X, Y, and Y. So this is what we have. And they say, describe how structures W, X, and Y are adapted for wind pollination. We can see that here, you can see this is the stigma, is really feathery. So W, which is a stigma, is feathery to catch the pollen. So that is an adaptation for wind pollination. When we see the anthers here, they are exposed, meaning the wind can be able to blow the pollen from the exposed anthers. And then here for Y, we can see here, this part here, this way, which is the filament. are attached to the anther. So the filament is not rigid. So this ensures that it can be flexible for wind to blow the whole thing in order for this parcel to occur more efficiently by the aid of wind. So the 3 would earn you 3 marks. Moving on down here, they say structures W, X, and Y are adapted for wind pollination. Give 2 other differences between wind-pollinated flowers and insect-pollinated flowers. Wind-pollinated flowers are not scented, and their color is dull, or the color of their petals is dull. It could be green. And then the pollen that they produce is going to be smaller and lighter, and they do not have nectar, because nectar is present if insects are going to pollinate in order for this nectar to be a reward to the insect pollinators. So let's continue. Here they say, wind-pollinated flowers often cause an allergic response in people. This is known as hay fever. Most people in the United Kingdom who get hay fever have the symptoms from April to September. They say the graph shows the changes in total pollen count for 3 different plant types from March to September during 1 year in the United Kingdom. So we can see from about maybe March to the end of May towards June, we can see there is more tree pollen. There is an increase in the concentration, and then it goes down a bit. And then from about this point here to this point here, we can see there is a lot of grass pollen. Finally, towards the end, we can see there is a lot of weed pollen. So it depends on which pollen somebody is allergic to, so that they can show symptoms in specific months of the year. So here they say, as part of an investigation into pollen allergy, 5 people keep a diary of their hay fever symptoms. They do this for the same year as the pollen count. The table gives the results. So we can see there is person A. They have severe symptoms in the month of April and May, meaning they are going to be allergic to this. They also show mild symptoms from March to June. So because there is a March to June, you can see still the same part, March to June. It means these people are only allergic to this part here, which is, you can see they're only allergic to tree pollen. Here we can see, yes, this is still in the area of tree pollen. And from July to September, they have no symptoms. So it means they will not be allergic to grass pollen as well as the weed pollen. So that's it. For person B, we can see person B, they say the severe symptoms are between June and July. And then the months with mild symptoms are March to May, meaning they have mild reaction towards this pollen and a strong reaction towards that part. So I said they are mildly allergic to tree pollen. Here, no months without symptoms. So it means they will also be mildly allergic to the weed pollen. However, they are severely allergic to grass pollen, as we can see. So in video C, symptoms from April to September. And then when they have mild symptoms is usually March. And here, there are no months without symptoms. So these are allergic to all types of pollen. And as you can see, it's from April to September. So all 3 types of pollen. Now, individual D, the months with severe symptoms are none. Months with mild symptoms, none. And then months with no symptoms, we can say all. It means they have no pollen allergies. Individual E, they say the severe symptoms are from June to September. So we can see it means they could be allergic to grass as well as weed. They also have mild symptoms from March to May, meaning they are mildly allergic to this one. So severely allergic to this and mildly allergic to this one. That is what we can see from the table. So let's continue to the next question. Here they say, using the data in the table and the information from the graph, discuss the likely cause of allergic reaction or allergic responses in each person. I've already said all these, but I'm going to go through them again. I said based on the data, a link exists between the pollen concentration and allergic reactions. Individual A is only allergic to tree pollen, as I showed you in the table, and individual B is mainly allergic to grass pollen and mildly allergic to tree pollen as well as weed pollen. For individual C, they are allergic to all of the 3 types of pollen, since they had symptoms during all months. Individual D has no pollen allergies. From the table you saw, they do not have any symptoms. Individual E is mainly allergic to weed and grass pollen, yet mildly allergic to tree pollen. And in conclusion, I say the data was carried out for only 1 year, so it may not be conclusive enough. Maybe they have to carry it out for longer periods of time. And if they found a way of including more individuals, then maybe the results will be more reliable. So let's move on. Here they say the allergic response to pollen is part of the body's immune response. Explain what is meant by the term immune response. Immune response, in this case, is a response to antigens or allergens that cause lymphocytes to produce antibodies and for phagocytosis to take place. Remember, in the immune response, it could be specific or non-specific. So this brings us to the end of question 5. Let's continue to question 6. Question 6. The diagram shows a plant cell in distilled water and a plant cell in a concentrated solution of sodium chloride. So this one here is in distilled water. We can see it's turgid. However, this one here is in a concentrated solution of sodium chloride, so we can see the cytoplasm is pulling away closer inside. So the first question they asked us is, which structure is labeled M? Structure M here, that should be the cell wall. The next, which structure is labeled N? N is the cytoplasm. Remember, this is the vacuole. That should be the cytoplasm. And if you compare this and this, we can see here the vacuole is kind of, the shape of the vacuole here and there is different because this one here is going to lose some water while this one here is going to gain some water. So the next page says, give the name of the liquid found in the gap labeled O in the cell in the concentrated solution of sodium chloride. Let us go back here. This is the gap here. We expect that to be sodium chloride solution. Next, they say explain the differences in the appearance of the cell in distilled water and the cell in concentrated solution of sodium chloride. So in distilled water, I said the water will enter the cell by osmosis. So from a higher water potential to a lower water potential, that means the cytoplasm will push against the cell membrane and therefore the cell will be turgid. It's going to swell up or be turgid. You could also reason in terms of the other cell that this part here is about distilled water, so we have to focus on that. However, if you want to reason in terms of the concentrated solution sodium chloride, you could do that as well. Next. Here they say describe an experiment you could do to show how different concentrations of sodium chloride solution affect the appearance of plant cells. Now here we set up 5 different experiments, and these experiments all have to have different concentrations of sodium chloride solution. That is how you get your first mark. Now we have to make sure that the plant tissues we are using are consistent. So the consistency could be attained by using plant tissues from the same onion. So here I said, immerse the plant tissues like onion epidermis into each solution. You have to ensure that the tissue is from the same onion. This ensures consistency. And then we have to let those plant tissues to be exposed to the solution for the same period of time. Then after we repeat the experiment for each salt concentration, this is going to ensure that we find the mean and then observe the tissues under microscope to confirm the changes. So whether it has become bigger or it has become smaller. And for each solution, we have to use the same volume of sodium chloride solution to ensure that they are all exposed to the same conditions. This brings us to the end of question 6 as well as the end of this video. Thanks for being with us. Please do not forget to subscribe to our channel. See you in the next video. Bye-bye.